Hello students, the topic for discussion of today is collection procedure of chordates and non-chordates. I am Dr. Bhagyashree Mohanto, Assistant Professor, Department of Geology, USTM, and this topic is for BSZ first semester students. First of all, we are going to discuss about the collection procedure of chordates, and our first phylum is Hemichordata. One of the example of hemichordate species is Balanoglossus. Here in the slide you can see the diagram of Balanoglossus species. They are basically marine, bilaterally symmetrical organism. Some of their characters are chordate-like and some of the characters are not. So therefore they are called as hemichordata, means half chordata. Such kind of specimens are collected from the seashore where they dug in the mud, and to collect them, specimens are dug from shallow water with a shovel, or they can sort it by hands. Then a large shovel full of mud which surrounds the worm is transferred in a bucket full of water. Then the mud is then submerged in the water and the specimen carefully taken out to avoid the damage. After taking the specimen, we can preserve them in chemicals like 10% formaldehyde or 10% alcohol, and then we can store them in our museum for future references. Our next phylum is Urochordata, and the tunicates are coming from this Urochordatan phylum. In the slide you can see the photograph of a Urochordatan species, that is tunicates. They are very beautiful creatures found in deep sea. They normally stay there either in different stones underwater, but during the summer month they come outside the sea and they can be found in the seashore, and at that time we can collect those tunicates or other Urochordatan species with the help of different kinds of nets like deep net, tow net, etc. In the diagram you can see the structure of a deep net, and with the help of it we can collect tunicates and then we can preserve them for future references. Our next phylum of interest is Cephalochordata, and Pyrosoma is one of the example of Cephalochordata phylum. In the slide you can see a glass-like animal in the sand. Basically it is from the picture is from the seashore, and they are basically marine organism coming out from the sea. And to collect such species we can use sand screening method with a coarse 1/8-inch collecting screen. So after collection, sand is washed through a screen and the animal collected in a bucket. Then dredge net is also used for collection purpose of this Cephalochordatan species, and after collection we can preserve them as different kinds of preservatives like I said earlier, formaldehyde, alcohol, etc., and then we can store in our museum. The collection procedure for fish are different because we have different kinds of water bodies in our world. Some of them are freshwater bodies, some of them are saltwater bodies, some of them are stagnant water, and some of them are free-flowing water. So in different water bodies, the physiological structure and the behavioral structure of fishes are different. So for collecting purpose, the fish gears, those are used to collect those fishes, are also different. So here in the table you can see different kinds of collecting equipments for different kinds of water bodies. First type of water bodies, that is estuaries, lagoons or backwater. We can use bag nets of different types. bamboo screen, purse net, seine net, drift grill net, leaf net type deep net, hand lining, etc. for collection of fishes. Then in ponds, zills, lakes and reservoirs, that is basically stagnant water, the collecting equipments like seines and drag nets, trap nets, scoop nets, gill nets are used. In hill stream fishes, various types of traps and cast nets are used. In rivers, seine nets and drag nets, purse net, drift grill net, dip net, bag net, trap net, cast net, hook and lines, etc. are used. And in marine water, that is in sea, rectangular or conical fixed net in the tidal region, then moving and fixing bag nets, seines, gill nets, drift nets, drag nets, cast nets, hand lines, long lines, such kind of fishing gears are used to collect those fishes. For collection of amphibians and reptiles, different kinds of collecting mechanism are used. The first and widely used collecting mechanism is hand collection, and for hand collection we do not need any kind of specialized equipment. It is easy. We can collect non-poisonous reptiles with the help of hand collection, and amphibians as well. So hand collection is one of the main mechanism of collection. Then netting is used. Netting means different kinds of nets are used to collect all those non-poisonous reptiles or amphibians. Then noosing is used. Noosing, you can see in the first diagram, first photograph. Reptiles are basically collected by noosing mechanism. Here one thread is used and a trap is formed with the help of a knot in the thread, and with the help of this knot we can trap amphibians or these reptilian species, and then we can collect them and then preserve them for our future references. Then another type of collecting is drift fences. It's like a fence, and with the help of this fence we can collect reptilians and amphibian species. Then different kinds of traps are also used. Here you can see 4 types of traps: wire funnel trap, sticky trap, cellotape trap, pitfall trap, etc. And in the picture you can see 2 kinds of traps. First one is pitfall trap, where an amphibians, basically amphibian species are collected by pitfall trap, where they fell down into the trap, and then we can collect them from this pitfall trap. Then sticky trap and cellotape traps are also used, where some sticky materials are used, where the amphibians or reptilian species pasted on there, trapped on there, and after that we can collect them. Then another types of trap is wire funnel trap. In this trap, funnel made from wires are used, like in the third diagram in the slide. Here amphibians or reptilian species can enter into the trap, but they could not come out from the trap. So for snake collection, 2 kinds of equipments are used, that is snake hook and snake tongue. As because snakes, some of the snakes are poisonous, so we cannot directly touch them with our bare hand, so we can use hook and tongue to collect those snakes. And catapults are also some equipments which is basically used to throw some subjects with it, and with that throwing we can kill different kinds of amphibians or reptilian species from a distance, and after killing we can collect them and then preserve them for our future museum and for our future studies. For collection of birds, we can use two types of methods. One is shooting methods and second is mist net. For shooting, we can use 410 shotgun method where we can shot our bird of interest from a distance and then we can collect them. Uh, and also we can use mist net. These mist nets are placed basically in the cropland and the birds are trapped with their wings in the nets as you can see in the diagram in the photograph, and after they trapped in the nest we can collect them and then we can preserve them. For mammalian species, there are lots of traps depending on the size of that mammal. For small mammals, different types of traps are there, and for large mammal, very complex types of traps are there. So first in the picture you can see snap traps. Such kind of traps are used for rodents, smaller mammals. Second is steel traps. This is a complex type of traps basically used in the jungle for elephants or bears. Third one is bottle traps, which we can make in our home also to collect different types of rodent species. We can simply made it from a simple bottle. Then harp traps are there. Harp traps are basically used for collecting of bats, and they attract the bats, and when bats entered into the trap, a bag is there and the bat is captured inside that bag without any harm, and with this process we can translocate bat from one place to another also. And the last one is snare, and this kind of traps are used to collect used to trap larger mammals like wolf, and then fox, dogs, etc. Now we are going to discuss about the collection procedure of non-chordates. The methods applicable to terrestrial habitats, now we are going to discuss. And the first kind of trap is light interception traps. Such kind of traps are basically placed in cropland, and it is used to collect different kinds of terrestrial insects flying over the croplands. In this trap, as you see in the diagram, there is a collecting tray, and in that collecting tray, ethanol or soap solution is there. And when insects flies over this trap attract those insects, and these insects just fell down in the collecting tray where preservatives are there, and those insects are directly preserved in that tray, and after that we can store them in our different kinds of bottles and store them in museum. Then a special trap is for different kinds of invertebrates like ants, Drosophila, moths, butterflies, etc. And such kind of traps are called as bait traps. So basically three kinds of baits are there: peanut butter bait, rotting mushroom, bananas or meat soaked in water bait, and third one is sugar bait. Peanut butter bait is basically used to attract ants. Second one, rotting mushroom, banana or meat soaked in water bait used for attract Drosophila, and sugar baits is used for moths, butterflies, beetles, dipterans and hemipterans. Peanut butter bait is basically made with small wooden ice cream spoon or chopsticks which are dipped in a peanut butter and placed at regular intervals, and ants are attracted with that baits comes there, and we can collect those ants from that place. Then to make rotting mushroom, bananas or meat soaked in water bait, the bait is basically smeared on small sponge or other absorbent material and placed on tree trunks or other substrate, and Drosophila are directly attracted there, and then we can collect them. For sugar baits, They are simply hanging containers with a combination of molasses or sometimes fruit juice or kero syrup. Then the mixture may be smeared directly onto the trees as well. And with this bait, moths, butterflies, beetles, dipterans, hemipterans are attracted there, and we can collect them from that bait. Now it is another kind of trap called as Burley's Dahlgren funnel. Here in the slide you can see the structure of this trap. In this trap there is a bulb in the upper side, and there is a collecting jar full of 70% ethanol in the lower side, and in the middle we can place the soil sample or leaf litter where invertebrate species must be there. So basically this kind of trap is used to extract invertebrates from soil and leaf litters, especially insect larvae, nematodes, isopods, slugs and snails, as well as arachnids. So what is the principle of this funnel is, when we open those bulb, switch on those bulb, due to the light and the heat, the invertebrates present in the soil sample or leaf litter sample, they go downwards due to the heat, and when they go downwards there is a collecting jar there, and in the collecting jar they fell down, and as because it is filled with 70% ethanol, those invertebrates directly preserved in those chemicals, and after that we can collect them and then store in our museum. Now another kind of trap is pitfall trap. Here a preservative called polypropylene glycol is used. Pitfall trap is also used to collect amphibians and reptile species, as I told you earlier. In case of invertebrate as well, pitfall trap is used to collect different kinds of invertebrates like spiders, ground dwelling beetles, oligocets, annelids, slug, etc. Now there is a method called chemical extraction method, where a solution of 2%, 0.2% formaldehyde is used. It is thoroughly applied on soil of desired area, and the invertebrates emerging out were collected. With this chemical extraction method, we can collect earthworms and larvae of many flies. Then another kind of method is flotation method, and in this method we can collect arthropod species with the help of hydrocarbon solvent, as we all know that chemical like hydrocarbon can wet the arthropod cuticle, and water cannot wet their cuticle. So a mixture of hydrocarbon like gasoline and water was made, and we can add the soil sample or leaf litter sample there where invertebrate species are there. Then these contents are allowed to settle. Then arthropods will tend to be found in the gasoline layer, which floats above the water, while soil, leaves, etc. will tend to be found in the water layer, and after that we can separate the gasoline layer and then collect the invertebrate species. Canopy fogging is another kind of collection methods. This method is basically used to collect different invertebrate species found in the canopies of large trees. From the bottom we use some fog, like you see in the first picture, and due to this fog the invertebrate species will fall down. In the ground we use a net, and in that net the invertebrate species are fall down, and after that we can collect them from this net. Bead sheet is another kind of collecting mechanism. Here we can use a sheet down a particular tree, and if we beat those tree with some stick, the all invertebrates present found in that tree will fall down in the sheet, and then we can collect those invertebrate species from that sheet. Now we are going to discuss about the methods applicable for aquatic habitat. So the first one is plankton net. Here in the picture you can see different types of plankton nets. Some of them are smaller in structure, some of them are larger. They are sometimes used in freshwater, some of them are marine water plankton nets. Here these plankton nets are basically used to collect different types of phytoplanktons or zooplanktons. The main net which is attached in the plankton net is made up of silk, organ dye, muslin, terylene, nylon or synthetic fibers, and with the help of it we can collect planktons, then store them in our museum. Then another types of machine is there, that is cylinder sampler. These cylinders of known size slowly lowered onto the water to collect crustaceans and planktons. So it is a machine and it directly collects different crustaceans and planktons from the water sample. Then water pumps and hose are used. It is used in quantitative plankton sampling. It is a pump with a rotary action and fitted with flexible inlet and outlet hose pipes that is generally used. The inlet pipe is graduated to show the depth of the vertical descent. A weight is also tied to the inlet hole of the hose, which makes the pipe easy to sink and keep it vertically straight in water. This method has certain disadvantages as the water flows from all the direction into the inlet hole at a particular depth. The microhabitat occupied by the plankton cannot be ascertained. So there is another kinds of collecting equipments to collect aquatic organisms, that is dredging method. Dredging is excavation activities carried out underwater. A fishing dredge is also known as scallop dredge, and this dredge is used to collect oysters, crabs, sea cucumbers, etc. Now we will going to discuss about some common methods. Those are used to collect terrestrial and aquatic habitat. First one is hand collection method. It is the simplest form of collection that is done by hand with small vials or collecting jars. It is advisable to carry different size of vials in a small bag. The greatest advantage of this method is that one can sample a very large number of different habitats in a short amount of time. The second common method for both terrestrial and aquatic organisms is floodlights, flashlights, and backlights. It is a novel way to capture nocturnal invertebrates. Nearly half of all invertebrates are active at night. A flashlight or headlamp is used to attract the insect which are collected by hand. Blacklight traps include a variety of trap types, but what they all have in common is the black light or ultraviolet light, and is used for collecting many insects that are active and flying at night and are attracted to UV light. Most likely invertebrates to be encountered includes knotweed moths, wolf spider, fishing spider, jumping spider, beetles, dipterans, and hemipterans. So these are the diagrams of different types of nets and collecting equipments. Those are used in both terrestrial and aquatic organisms. First one is aerial net. Aerial net may be used in the in water also. Aspirators and vacuum devices, these two kinds of devices may be used in both terrestrial and aquatic organisms. Then emergence traps are there in the diagram. You can see these emergence traps we can place in the crop field where lots of water and vegetations are there, and we can trap many insects and organisms, both aquatic and terrestrial habitat, with the help of this trap. Then sweeping and netting method is there, and this sweeping and netting method is basically used for terrestrial insects. So dear students, this is all about the today's topic, that is collection of methods of collection of chordates and non-chordates, where we include both aquatic and terrestrial organisms. And to make this PPT, I have consulted a very beautiful book, that is Animal Taxonomy, and this Animal Taxonomy book is written by Monidip Raj. Dr. Monidip Raj was a renowned taxonomist of Assam, and then for this picture courtesy for to collect those photographs, I have consulted slideshare.com. So that's all for today. Thank you very much.